So you guys had your midterm on Monday, and I've got those graded, and maybe you've seen the scores online, by and large, pretty well. And I'll pass those back to you for you to look at today during the laboratory period. On the Friday quiz here in lecture will be on, boy, what did we just finish? Signal transduction, thank you. It'll be on signal transduction, but I'm also going to ask a few questions on this chromosomes chapter that we started with and are going to continue with today. Alrighty, well, I guess I mentioned last time that the focus of the course is going to shift a bit now, and we're going to spend pretty much the rest of the semester talking about DNA and genes and how they work in a lot more detail than we covered so far. And what I wanted to do was just give you some background of how people discovered that DNA was the genetic molecule, because for a long time they didn't know that. And so I guess I covered most of this on Monday, but let me do a quick recap of it. Probably the first person who we should mention if we're talking about genes is Gregor Mendel. And remember, he was an Austrian monk who got interested in how traits are passed from one generation to the next. And so he worked out some rules of genetics, essentially mathematical laws that predict how traits can be passed from one generation to the other. And I think I showed you this slide here, where he would use pea plants and cross them, breed them with each other. And he found some interesting results, like a purple one and a white one crossed gave all purple in the next generation. But if you mated that purple generation with each other in the generation following that, you got generally 1 out of 4 having the white trait that you saw in the original generation back there. There we go. And 3 out of 4 would have the purple trait that you saw there and there. And anyway, so he worked out some mathematical rules, which are called Mendel's laws of genetics. And that we'll study these actual rules in a couple weeks, but the point I'm making here is he understood the rules that genes were passed from one generation to the next, but he didn't know what a gene was. To him it was just something, some kind of molecule, he didn't know what, that controlled a trait and was passed from one generation to the next. The first hint of what genes were actually made of was discovered by this man right here, Walther Fleming. He was a German scientist, and he discovered chromosomes by looking at the nucleus of cells. And he didn't know what chromosomes were, what their functions were, or that they contained genetic material. He just said there's something inside the cells that in the nucleus that's called a chromosome. But a few years after that, these two gentlemen, Walter Sutton and Theodore Boveri, and a few others, noticed that there were some similarities in the number of chromosomes that they could see inside various cells, and these rules of genetics that Mendel had talked about. To be a little bit more exact, Mendel had predicted that all organisms have 2 of each gene, but when they make their sperm or their eggs, they only put 1 of each gene into those reproductive cells. And what Sutton and Boveri observed is that the number of chromosomes in most of the cells, the body of 2 of each chromosome, but in your egg or your sperm, you only have 1 of each chromosome. And they said, well, that's exactly what Mendel described as the rules of genes. And so maybe the genes are on the chromosomes. That was the first suggestion that genes might hold chromosomes. The first direct evidence of that was by this man right here, Thomas Morgan was his name. He was an American researcher. He was studying fruit flies, and he found a specific fruit fly mutation, which is a genetic trait. It's carried by a gene, where instead of the normal red color of the fruit fly eye, it was a whitish color. And what he found is that the offspring of this mutant fly, all the offspring that inherited one chromosome, one specific chromosome from that fly, always got the white eye trait. And so that that strongly suggest that indeed the genes are on the chromosomes. Genes are part of chromosomes. So there was a lot of interest in exactly what the structures of chromosomes were. And here's a cell with some chromosomes inside of it. You can see them under the microscope as little dark thread-looking structures. Diagram-wise, they look like this. The dot, the green dot in the middle is the centromere, which remember is kind of a handle that the cell can use to move the chromosome around. And despite the fact that they call it a centromere, it's not always right in the very center of the chromosome. The part that I'm showing in red there represents the big piece of double-stranded DNA, which is really, well, we know now that's where the genes are located. But anyway, a long piece of double-stranded DNA. But that DNA is wrapped around some proteins called histone proteins, which are the blue dots in this diagram right here, just to condense the chromosome to make it smaller. And for decades, after scientists accepted the idea that the genes were on the chromosomes, they thought the proteins were the genes, not the DNA. And it kind of makes sense because traits are complex things, and proteins are more complex than DNA. Proteins have 20 monomers, DNA only has 4 monomers. Proteins can carry out enzymatic functions, they do things, whereas DNA by itself in a test tube doesn't really have any activity. And so people just assumed that the proteins were the genetic molecules. But eventually some evidence came along that correctly showed that DNA was actually holding the genetic information. And the first person who found it was the DNA was a researcher named Oswald Avery. But he was actually, when he made his discovery, he was actually extending the work of another researcher named Frederick Griffith, who was this gentleman right here. And so here's what this Griffith fella did. He had 2 strains of bacteria, one virulent and one non-virulent, one disease-causing and one non-disease-causing, in other words. But he found if he killed these ones with heat, which basically tore them apart into their smaller molecules, and then allowed some of those molecules from the killed-off heat bacteria to be transferred to the non-virulent bacteria, it would take on the traits of the other one. In other words, it would become a virulent bacteria. So some gene, some molecule that carried, that affected traits, was able to pass from these dead molecules from the heat-killed bacteria to the non-virulent bacteria and transform it. But Griffith was not able to ever find out exactly what that molecule was. The laboratory techniques at that time were just not up to it. And that's where this Oswald Avery fella came in. He repeated Griffith's research, but he was able to do some analysis on exactly what molecule was passing from one bacteria to another. So he did the same thing. He killed off the virulent strain and analyzed what molecule was moving between them, and he found out that it was DNA. So DNA, he concluded, must have been what's carrying the genetic information to transform, to pass traits from this dead bacteria to this living bacteria right there. The only problem was that he was not able to get 100%, when he was analyzing the molecule that was passing between, he was not able to show that it was 100% pure DNA. There were still a few proteins, histone proteins, that were in it. And so there was such a strong bias among scientists at the time that proteins were the genetic material. A lot of people said, well, it's still the proteins in your sample there that are carrying the genes, not the DNA. So it wasn't a slam dunk, for so to speak, for DNA being the genetic molecule. But the actual proof that pretty much all biologists accepted that DNA and not the proteins were the genetic molecules came about 10 years later from these 2 researchers. Ooh. There we go. The woman's name is Martha Chase and Alfred Hershey. is his name. And they did what's called the Hershey-Chase experiments. And they did some research on viruses. And I think I showed you these slides last time. Viruses are extremely simple organisms. They have a very, very tiny chromosome, the red line here, and it is very small. It just has maybe on the order of 10 genes. And the viral chromosomes don't have any histones, so it's just a raw, naked piece of DNA. And surrounding and protecting the viral chromosome is a protein structure called the capsid, which is the blue dots right there. And it's basically just a protein shell to surround and protect the chromosome. Now, some viruses, the capsid has a downward extension that looks like this, that's also made of protein. So don't let that distract you from the basic simplicity of the structure. It's still just a protein outer part and a DNA chromosome with no histones on it. Viruses are such simple organisms that they can't reproduce by themselves. They don't even really have a metabolism by themselves. The only way they can get reproduced is by squirting their genes inside a cell, a living cell, and that fools the cell into reproducing the virus. Here's one right here. The virus has landed on the surface of this cell. Oh, come on, laser pointer. The virus has landed on the surface of this cell right here. It squirts in its genes, and to the cell doesn't know those are viral genes, it's just some more genes to the cell. So the cell says, okay, I'll express those genes. And of course, that's bad for the cell because those genes encode more viruses. And so the cell becomes a factory for new viruses, which eventually burst out of the cell, killing off the cell. And now these viruses can spread to other cells, and that's how the infection propagates itself. Well, when Hershey and Chase started their work, people didn't know that it was the DNA that was getting inside. They knew the basics, that the virus is attached to the outside of a cell and squirt something inside, but people didn't know whether it was the DNA getting inside or if it was the protein part of the virus getting inside. So up to that point, they really couldn't use viral research to clear up the question whether the DNA was the genes or the protein was the genes. Well, that's the question that Hershey and Chase addressed, and here's how they did it. Let's see. They were able to make a virus that had radioactive proteins. In other words, a radioactive capsid, which is the red part of the virus that you see here. Whereas the DNA was not radioactive in their experiment. And then they allowed the virus to infect some cells just for a few moments, and then they threw the whole thing in a blender. And what the blender did is it knocked off the parts of the virus that had not gotten inside the cells. In this case, the capsid, the protein coat. And when they did this, they found that the infected cells were able to make more viruses. In other words, the genes must have gotten inside, even though the proteins never got inside. And so that is essentially evidence that the proteins are not the genetic molecule, but that the other part of the virus, the DNA, was the genetic molecule. And to prove their point conclusively, they did essentially the opposite experiment. They now made some more viruses that had radioactive DNA, not radioactive protein. And when these viruses were allowed to infect the cells just for a few minutes, and then they put them in the blender to knock off the parts that didn't get inside the cells, they found that the radioactive material, the DNA, was what got inside the cells, and that was able to direct the cell to make new copies of the virus. So it proved very conclusively, or they proved very conclusively, that it was that DNA is the genetic molecule, not the proteins themselves.
When they blended that, the cells didn't get damaged?
You would think that the cells would be torn open, and yeah, that would be a problem in their experiment, but I think they blended it at such a low speed that almost all the cells were left intact. Yeah, that's a good observation. All right, so at that point, after the 50s, biologists accepted that DNA was the genetic material. And as I said, that surprised a lot of people because DNA seemed so simple. It only had 4 monomers, and they wondered, well, how can something so simple carry all the genetic information that's necessary for very complex life forms like ourselves? Well, so the focus was then attention was given to DNA to try to figure out how it worked. And to begin with, no one really knew the structure of DNA. They knew that it had G's and A's and T's and C's that were somehow connected to each other, but they had no idea that it existed in this double helix of complementary strands. People began to try to figure out what its exact structure was. This gentleman here is named Linus Pauling, and he did some investigation to try to figure out DNA structure, but he did not get it right. He had some molecular modeling kits, some little balls that represented carbon atoms and nitrogen atoms and oxygens and hydrogens, and he tried to assemble what he thought the DNA structure was, and he came close. He came up with a triple helix model, which we now know is not accurate, but